Hello, in this video we will be going over some applications of the reproductive cloning in mammals. A brief summary of what reproductive cloning is. It is a process of producing an identical copy of an entire organism. This is possible through artificially induced asexual reproduction. Keith Campbell, Ian Wilmot of Roslin Institute, with many other scientists, performed the first ever successful cloning of animals for the first time in history. The most well-known example is Dolly the sheep, which we will be discussing in this video. Dolly was born on the 5th of July in 1996, and she was the first mammal to be cloned successfully. Dolly is here on the left. Standing beside her is her surrogate mother. So the question is, how does this reproductive cloning in mammals work? Let's discuss how Dolly was brought into this world. First, a mammary cell was taken from the mammary glands of sheep number 1, which was a 6-year-old fin-dorsate sheep. Scientists found a way to reprogram the mammary cells to ensure they are alive and also to stop further growth. The nucleus from this cell was then separated and the cell was starved in order to force it into an almost embryonic state. Then the scientists took an egg cell from sheep number 2, which was an adult Scottish black-faced ewe, the egg cell donor. The nucleus in this egg cell was then removed, and the nucleus from sheep number 1 was then inserted into the now empty and fertilized cell of sheep number 2. To make sure the two cells were fused together, electrical pulses were used to stimulate fertilization. The fused cell was then allowed to develop. It was allowed to develop into an embryo. The developed embryo was then implanted into a surrogate mother, which was another Scottish black-faced ewe, similar to the egg cell donor, sheep number 2. This implanted embryo was allowed to develop naturally, and over a period of time, Dolly the sheep was born. Scientists actually fused 277 cells, and one developed into a full adult, which was Dolly here. Although Dolly was given birth by a surrogate mother similar to sheep number 2, a Scottish black-faced ewe, Dolly surprisingly had all the characteristics of sheep 1, the adult fin-dorsate ewe. Dolly's characteristics were identical to those of sheep 1 due to the fact that the nucleus was taken from sheep 1. Since a nucleus contains all the chromosomes of a body, these chromosomes determine the characteristics of an individual. Therefore, the Dolly, the sheep, got all the chromosomes from sheep 1, and that is why it was identical to sheep 1. Dolly was actually a very healthy young sheep and lived at the Roslin Institute, and during her time there, she was able to produce an offspring, and at the age of 6, she was unfortunately had to be euthanized due to lung disease complications on the 14th of February 2003. This successful cloning of Dolly brought revolution in the field of reproduction by cloning. Since then, several attempts have been made to produce cloned animals, but many of these animals were found to be severely diseased. Some died at birth or a few days after birth. In terms of visual looks of the cloned animal, it will not completely look like the original animal. This is because in conjunction with the genetic material, the environment also plays a large role on how an organism develops. This can be seen on the first cloned cat, who was a female calico cat that is very distinct from her mother. Rainbow here on the left was the nucleus donor, and the cloned cat, whose name was CC, short for carbon copy, is here on the left. The difference in color and pattern of these cats cannot be attributed exclusively to genes and thus the result of epigenetic and environmental factors. That will be all for this video, and thank you for watching.